Good afternoon and welcome to the Wednesday afternoon lecture series. We are particularly pleased to have Dr. Peter Agri, a Nobel laureate, speaking at this event after our immunohematology and blood transfusion symposium this morning and this early afternoon, and tomorrow's red cell genotyping symposium, which will also take place here in the Mazur Auditorium. Now, Dr. Agri, as everybody of you will be aware, shared the 2003 Nobel Prize in Chemistry for discovering the Aquaporins, a family of water channel proteins found throughout nature and underlying numerous physiological processes and clinical disorders. And this goes back to the late 80s. And when I did my postdoc in San Diego in the Department of Biology as an MD, it's a tough time. You might understand that the PhDs looked a little bit down to us on the MD side. Well, at any rate, in this Department of Biology, several laboratories in San Diego, UCSD, worked already on the water channels. It was a momentous discovery, and it came about serendipitously by originally studying RH, the blood group system, and a contaminant, and that turned out to be another blood group system, Colton, and this was the sought-after water channel where people in biology sought after for 100 years. So it's really an enormous achievement, and we're so pleased to have Dr. Agri here. Later, he became deeply involved in multiple global issues as appropriate with his track record. He currently directs the Johns Hopkins Malaria Research Institute, leading field research in Zambia and Zimbabwe. As chair of the Committee on Human Rights of the National Academies, Agri led efforts on behalf of imprisoned scientists, engineers, and health professionals worldwide. He is past president of the American Association for the Advancement of Sciences. Agri leads scientific diplomatic visits and meetings with leaders of countries including Cuba, the Democratic People's Republic of Korea, so that's North Korea, Myanmar, Burma, and Iran. We are really pleased to have you here. Looking forward to your presentation, and I cannot tell you how excited I am to listen to your record on these originally RH research, and you got the better end of the RH research.
Good afternoon. It's a pleasure to be here. I've been looking forward to this, and I'm a little intimidated speaking to the transfusion medicine crowd, but I started as one of you and branched out as nature serves some things our way. So I'm going to talk about water channels, but I'm going to do so in the context of kind of the long story from the beginning up to the present. And there's a lot of things that I will not be touching on, but in most cases I'll be talking about the work of an individual young scientist. So you young scientists here, this is particularly pitched to you. The work that you're doing right now will have consequences years from now. It's hard to foresee how far things can go, but have fun, and I hope you aren't disappointed in my lecture. So water, ubiquitous on Earth, essential for life, the solvent of life, according to Albert Szent-Györgyi. But water brings problems when it's in disequilibrium. Tranquil rivers, when they descend through rapids, become torrential. The changes in global water status, the Katrina disaster, the tsunamis, the melting of the polar ice cap, all are having global consequences. But in biology, water is also important. And it's something I really didn't pay much attention to when I was a student, taking physiology courses. The notion that salt had specific transporters seemed clear, but water just sort of went where it needed to go. But it turns out there's a special plumbing system, if you will, for water. And that's what I'll be talking about, the Aquaporins. So the background preceding our entry into the water channels is represented in this slide. On the left, you'll see a simple membrane bilayer, which is permeated by diffusion, simple diffusion of water molecules back and forth across the membrane. But it's clear that there are some tissues, some cells that have very high water permeability. And these were recognized to have the need for special water selective channels, renal tubules, secretory glands, and red cells. The difference between diffusional permeability and the channel permeability was one of magnitude, the diffusion being a low capacity bidirectional movement, whereas the putative water channels were thought to be specific for water. Not even protons could pass them. And the movement was directed by the osmotic gradients. So this process of osmosis that we all learned about as children in school occurs in some tissues very rapidly, rapid osmosis. Now in 1970, a very important experiment was reported by Robert Macy at the University of California, Berkeley. A physiologist was looking at water transport in red cells, and he went through the chemical catalog, the chemicals on the shelf, and he discovered mercuric chloride would stop water transport in red cells. And he then washed the cells free of the mercuric chloride treated with a chemical reducing agent. He could restore water permeability. He could turn it off and on. And he predicted there must be water selective channels, but neither he nor other investigators were able to identify it for a variety of technical reasons. So the observation of the first water channel, which we now call Aquaporin-1, was a sheer serendipitous observation. As Dr. Flegel said, my initial work as a hematologist in a very small lab at Johns Hopkins was on the biochemical nature of the Rhesus blood group antigen. In 1982, Stephen Moore in Edinburgh, Carl Gomberg in Finland independently demonstrated that they could surface label red cells from RHD positive individuals and label a 32 kilodalton band, which did not label an RHD negative red cells. And by immunoprecipitations, they confirmed this is a part of the RH molecule. And based on that, we got going to biochemically purify this 32 kilodalton RH molecule, and we succeeded. And we looked at red cells from all different kinds of people, as well as other vertebrates. And we found that it was a protein shared in all species. It's an antigen in humans, but the core protein complex is apparently present in all species. Now, what we saw in the initial sodium dodecyl sulfate polyacrylamide gels shown here, and this is stained by silver, I should say, is a clear 32 kilodalton band and some slightly smaller bands in each species, which we dismissed as probably degradation products. By Kumasi blue, nothing stained at all. You couldn't see any of this. So we were hot in pursuit of the Rhesus blood group antigen, but this second protein fell in our laps. And we sooner or later figured that the second protein was not a breakdown product, but a contaminant. What a lowly start for a scientific project, a contaminant. But it was very abundant. It had interesting N-terminal degradation sequence. So we decided we'd clone it out. If we got the cDNA, that would show us homologous, and then we would know what it did. And the deduced sequence showed a 6 bilayer spanning membrane protein, configuration shown here, with an interesting internal repeat. The amino terminal half and the carboxy terminal halves of the protein are inverted with respect to each other. And the genetics database did have several homologs, but none of them functionally defined. Protein from the lens of eye, major intrinsic protein, a protein from the brains of insects, a protein that allows bacteria to use glycerol as a carbon source, and some gene fragments from plant tissues. So we were stuck. We didn't know the function of this new protein. And as biochemical investigators, I think you're all aware that a protein without a function is a scientist without a grant. And without a grant, well, it gets bad. There's great impetus to figure out things in science. But I talked to all of my colleagues, and they had interesting suggestions, but didn't quite fit together. So we did what any family would do. We went on vacation. Now, as scientists, we love our work, but not necessarily because we take home such large paychecks. Our family vacation every year was in a tent. We went camping in the national parks. And the kids loved that. We did this every year. We went to the national parks, went camping. And after doing this a few years, Mary said, my wife said, let's ask the kids which national park they want to go to next year. We'd been to Yosemite, Grand Canyon, Yellowstone. So we asked the kids, and they said Disney World, which is not a national park. And here are the little darlings. So we took them up on it. We went to the Everglades, and then we went to Disney World. And on our way back to Baltimore, we stopped in Chapel Hill, North Carolina, where I'd spent 3 years as a postdoctoral fellow. This was a wonderful time. Francis Collins was a medical resident at the hospital. It was a really high-energy, wonderful environment. And I became good friends with John Parker, a hematologist. And I stopped to see John. We're driving back to Baltimore. We took the break in Chapel Hill and told him about this new protein that I was convinced would be interesting, but I just couldn't figure out what it could possibly be doing. I asked him if it might be an oxygen channel. John looked at me and said, you know, I think this might be the long-sought water channel, something physiologists have been searching for a century. So really, the credit for the idea goes to John. And based on his suggestion, we pursued this. We collaborated with Bill Gugino at Johns Hopkins. And we expressed the new complementary RNA in frog eggs, Xenopus laevis oocytes. They have a very low inherent water permeability. They're about 1 millimeter in diameter, ideal expression system. So as you can see, the oocyte on the left and the oocyte on the right are pretty comparable. The one on the right is a test oocyte. The one on the left is a control. So the one on the right has been injected with 2 nanograms of the complementary RNA, cultured for 3 days in isotonic saline, isotonic buffer, no difference. But when transferred to distilled water, an immediate apparent difference. The test oocyte has exploded, producing much jubilation in the laboratory. This was Gregory Preston, the postdoc who did these first studies. And to tell you the truth, I took this snapshot of Greg 3 years after our first report. He was still celebrating. I think the discovery is the reward in science. But we really didn't know so much what we were getting into. But we had good friends. Joe Handler from the Heart, Lung, and Blood Institute had relocated at Hopkins and introduced me to Mark Knepper. And we suddenly had this group of really wise transport physiologists to advise us in our work. And so we proceeded diligently. And we did so by collaborating. One of the first challenges was to solve the structure of the aquaporin molecule, Aquaporin-1. We did that in collaboration with Yoshinori Fujiyoshi from Kyoto and Andreas Engel from Basel, Switzerland. And we got a high-resolution structure, which gave us a good idea of the function of the molecule. On the left panel, you can see one subunit. The protein is tetrameric. When viewed from the top of the cell, there's a single pore down the center, 3 angstroms in diameter. And in the right panel, we have a cross-section. So the extracellular surface would be above, the cytoplasmic surface below. A single channel, top to bottom, allowing water to move from an extracellular vestibule to an intracellular vestibule. Now, water is an interesting chemical, an interesting molecule, because it doesn't really exist as an isolated H2O molecule. It exists in a consortium due to hydrogen bonding, H2O to the nth power. So in the extracellular and intracellular vestibule, water is in bulk solution. But in the channel, the structure, shown just diagrammatically here, allows transport of water in single file with barriers to the movement of charged ions and a hydrogen bonding event at the very center. I'll point it. These 2 repeats that I mentioned briefly, this water molecule is isolated from the column of water for 1/1000th of a second, enough to prevent passage of protons. And it's very efficient. And this is work of David Cosimo, a graduate student who's now a faculty member up at Harvard. Multiple laboratories got active in the aquaporins, and together a series of 13 human homologs have been cloned out and hundreds of homologs from other species. Basically, every living organism has 1 or more aquaporins. And they exist in 2 basic forms. Highlighted in blue, the aquaporins are water selective. And highlighted in yellow, the homologs with slightly different structure are permeated by water plus glycerol and urea and other small uncharged solutes. We call those Aquaglyceroporins. And I'll tell you about each of several of these. So Aquaporin-1, where does it exist in kidney? We knew it was in the renal tubules, but we weren't very sophisticated. But we had the good fortune through Mark Knepper of meeting Søren Nielsen, a Fogarty Fellow here at the Heart, Lung, and Blood Institute, working on renal mechanisms. And together with Søren, we worked for a decade on a variety of projects, the first being the location of the Aquaporin-1 molecule in the kidney. And as you can see, the nephron unit shown on the left side of the diagram is highlighted. After passing through the glomerulus, the filtrate passes through the proximal tubule and the descending thin limb. And that's highlighted in green because that's where osmotic water permeability is constitutively high. This is a logarithmic compilation on the right. So high water permeability. Then the ascending thin limb and thick limb, negligible water permeability, emptying into the collecting ducts where water permeability is regulated. Vasopressin, high water permeability, lack of vasopressin is reduced. So the Aquaporin-1 protein is clearly in the proximal nephron, in the apical brush border, and in the lateral and basolateral membranes as well. You can see the gold dots from Søren's beautiful immunogold labeling. Intense presence of this protein in the apical surface. So it explains how water transits in transcellular fashion through the proximal nephron, going from the primary urine above through the apical surface, through the cytoplasm and out through the basal and lateral membranes, following the osmotic gradient created by salt and sugar transporters. Notice the tight junctions prevent movement of water from between the cells. So being a medical doctor, of course, we're very curious. What is the clinical significance of this protein? And we imagined that it would be catastrophic. Of course, we were wrong. This is where the blood group antigen work became very interesting. Because as transfusion medicine specialists, none of you will be surprised to think that a red cell membrane protein could be an antigen. You would assume so. Outside of the field, that's not so well understood. But when we did chromosomal localization of the gene locus, this is before Francis and the genome had been solved, we did fluorescence hybridizations. It turned out that the Aquaporin-1 structural gene lit up on the short arm of human chromosome 7. Now, the compilation in the McCusick Atlas didn't say anything about blood group antigens, but I happened to notice when I was visiting the New York Blood Center, my friend Colton Redmond, on the wall of his office was a human chromosome 7, with on the longer arm, an arrow pointing KAL. He was studying the KAL antigen. And in the short arm, there was an arrow pointing, it said CO. I said, Colton, that's very interesting. What is that CO on the short arm? He said, that's the Colton blood group antigen. And he started to explain about it. It's a protease-resistant antigen, a minor blood group. No one knew what it was. That was a very important conversation we had with Colton. In fact, I would like to propose we rename Colton the Colvin antigen. So based on that discussion, we contacted David Anstey and his team in Bristol, England, the International Blood Group Referencing System, which is a wonderful collaborative organization. And we were able to obtain blood, urine, DNA from a variety of individuals with different Colton phenotypes. And first we confirmed that the Colton polymorphism was a simple substitution, alanine-valine substitution at the 45th residue, which is an extracellular location. But that's not too important since almost everybody is Colton A or Colton AB. But in the whole world at that time, only a half dozen Colton null individuals had been known. So they, of course, became immediately interesting to us. And through our colleagues at the International Blood Group Referencing System, we were able to track down 3 different individuals, get them to come to Johns Hopkins for clinical phenotyping. I mean, it's amazing the power that science has when people work as colleagues. So shown here with permission is a Colton null individual, shown in the center, a retired school teacher from the south of France. And alongside her is a French-speaking postdoc, and Landon King, a postdoc in the lab, who did a number of phenotypic studies. And basically, everything he tested in kidney turned out normal. We're convinced there was going to be some kind of a clinical phenotype, but nothing turned out until he did the simple overnight thirsting. And we go to bed and sleep 7 or 8 hours. Of course, we drink no water. In the morning, we empty our bladders. And the last few drops of urine are highly concentrated because we send out vasopressin to conserve water during sleep. So in the morning, when we awaken, our urine tonicity will be up around 1,000 milliosmolars. But the Aquaporin-1 null individuals, and they all are homozygous for different knockout mutations, they're stuck at about 400 milliosmolar. They can't concentrate any further. So in terms of normal life with free access to fluids and air conditioning, they do fine. But if they were thirsted longer, they would clearly get in trouble. So it's a distinct form of nephrogenic diabetes insipidus. Now, Landon is a pulmonary specialist and looked at aquaporin distribution in the pulmonary microvasculature. And here's a nice micrograph that Søren Nielsen has prepared of a human capillary. And what you should see is that there's intense labeling of both the luminal and the abluminal surface. Capillaries are freely permeated by water. And in the second phenotyping, he evaluated pulmonary capillary water transport. And he did this at Johns Hopkins using a high-resolution CT scan of the lung, a method developed by a young scientist at Johns Hopkins named Elias Zerhouni, who had come from Algeria. I think you know him. He worked here for a while. So using Elias's technique, we evaluated water transport in the lung. And these are images from normal. These are the interesting ones. So what I'd like you to see is that at baseline, bronchioles have a thin wall, air space, venules next to it. And after infusion of 3 liters of physiological saline, what I'd like you to see is that the venules have now become expanded. And this is true for Aquaporin-1 null as well as normals. But the release of fluid from the venules to the peribronchiolar space, which normally occurs, it looks like this, so it goes from here to here, does not occur for the null individuals. There's a barrier to the entry and release of water from their vascular space. Now, we don't know the profound clinical consequences of this, but at birth, of course, the lung turns from a secretory organ to an absorptive organ. And this delay may be one of the reasons this is such a rare phenotype. We're still not sure about that. So a lot of interest was expressed by physiologists around the world. Actually, sometimes there was so much interest I felt a little overwhelmed. It was Mark Knepper that was on us right away. He was very pleased that we got the new water channel from the proximal nephron, but the collecting duct is the important part of the kidney. This is where vasopressin secreted causes water absorption to be increased. So a second member of the family is cloned by Sai Sasaki and his team from Tokyo. It's referred to now as Aquaporin-2. It's in the collecting duct. And it's in an interesting distribution in collecting duct. Unlike Aquaporin-1, it's at the apical surface. It's in intercellular vesicles in the pre-vasopressin state. And after infusion of physiological levels of vasopressin, it's at the apical surface. This is a classical paper which Søren Nielsen, Mark Knepper, and their colleagues published here 10 years ago. Hard to imagine, 10 years already, Mark. Excuse me, 20. Thanks. When you get to be my age, the numbers get hard. 20 years. That means we're 66. All right. Enough of my doddering. So the Aquaporin-2 moves to the apical surface. Water can then enter. But how does it exit the collecting duct principal cells? And that, interestingly, is due to other members of the Aquaporin family, Aquaporin-3 and Aquaporin-4. So it gets a little complicated. Now, clinically, this is really important. Inherited defects in the Aquaporin-2 gene cause a severe form of nephrogenic diabetes insipidus. This was work published by Peter Dean and Carl Van Oos from the Netherlands. Fortunately, it's very rare, but acquired defects are very common, with overexpression being found in situations such as congestive heart failure, leading to massive fluid retention, and underexpression, of course, in polyuria states such as bedwetting. So it's a very important molecule. I'm going to skip quickly through the family, and I'll just tell you a little bit about each of the other members of the gene family and who did the work. Aquaporin 0, also known as MIP, major intrinsic protein, is present in the lens. It's the source of, it causes cataracts in small children, mutations in the gene. And this is two different families reported by our colleagues from London, Shomi Bhattacharya and his team. And while you can't see anything from the visual exam, the cataract has multiple opacities in this mutant family and a single central opacity in this mutant family. So a loss of vision nonetheless. And it's believed that destabilizing mutations will probably be predisposing factors for the appearance of cataracts in older individuals. Another member of the family, Aquaporin-1, cloned from brain, expressed in an interesting distribution in the perivascular astroglial end feed. So the capillaries in the brain are, of course, surrounded by a parasite with a basement membrane, and then the astroglial end feed, these appear like suction cups here, provide support and a barrier to the movement of fluid from brain, from vascular space to brain parenchyma. And this is exactly where Aquaporin-1 is localized. The beautiful micrographs prepared by Søren Nielsen at the perivascular membrane, but not at the other membranes. Very special location. Now this location, of course, is the blood-brain barrier. And it predicted that defects in the causing brain edema would involve the pathways with Aquaporin-1. And that was first confirmed by our colleagues in a group led by Mahmoud Amiri Moghadam, a young Iranian scientist working in Oslo, who looked at two different mouse groups, normal mice after a defining brain injury, the injury being a middle cerebral artery occlusion, sustain a fair degree of infarct and edema. But the mutant mice, and the mice that he studied, had a defect in a bracketing molecule. The Aquaporin-1 is normal, but it's present in the wrong locations, the alpha-syntrophin. And these mice, when sustaining the same brain injury, have much less edema and infarct. And this was independently reported with other mouse models by groups in San Francisco and in Hungary. And it's clear that inhibition of the Aquaporin-1 molecule in terms of primary function or location would be of benefit for individuals developing brain edema. And brain edema is a tragic and very common problem. Stroke is the third leading cause of death in the United States. And in many cases, the individual with a stroke succumbs not to the infarct itself, but to the brain edema. This is also a big problem in the management of brain tumors. This is an MRI from a 65-year-old lady who had a large meningioma. And in the water-weighted image, you can see that around the meningioma was a massive amount of brain edema. Surgically, the meningioma was removed. This is the next day, but the brain edema persisted. It took months for that to resolve. This is a big problem. And this is a problem I know well because this was actually my wife, Mary, last year. She had a complete recovery. She's doing just fine now. But the management of brain edema affects people all around us. It's a big problem. Another member of the family, Aquaporin-1, is present in secretory glands, lacrimal glands, salivary glands, sweat glands. It's the last membrane water crosses during the generation of sweat, salt, and saliva. Now, a very simple and very elegant study was reported by Søren Nielsen and his colleagues in 2002, where they compared wild-type mice to Aquaporin-1 null mice created by Anil Menon and his colleagues at the University of Cincinnati. So the experiment, the test mouse, the paw was coated with starch and iodine. The animal is injected with pilocarpine. So amylase in the sweat digests the starch and it turns blue. So you see here all of the functional sweat glands. This mouse has sweat glands, but they're hypofunctional. The mouse can't sweat after pilocarpine. Now that may not be of such importance to mice living in cages, but it certainly has a lot of importance in terms of human physiology. In the summer of 2003, Western Europe experienced the worst heat wave in recorded history, temperatures over 100 degrees Fahrenheit for 2 weeks in succession in Paris. 15,000 unexpected deaths, individuals in their 50s, 60s, and 70s who were doing all right but didn't survive. And it's our belief that the aquaporins are part of the explanation for stress response. As we age, some of these pathways decline, sensation of thirst may be diminished, the ability to concentrate urine diminished, the ability to sweat diminished. And any other factor such as infection might be enough to put people over the edge. So it's probably well taken that the aquaporins serve as environmental response modifiers. Now I'm going to talk briefly about the glycerol transporting homologous, which we call the Aquaglyceroporins. And they're structurally very similar, but they're not identical. In this slide I borrowed from Bing Yap at the University of California, where he's juxtaposed the pore lining residues in the water channel from mammalian red cells. Notice the arginine is present. The darker shading represents the residues in the water channel, and the lighter shading represents the corresponding residues in the glycerol transporter. And what you can see is the cysteine, the mercury inhibitory site, is replaced by a phenylalanine, a hydrophobic trap door. This large histidine, which provides a positive charge, partial positive charge here and here to help repel protons. This histidine is missing in glycerol transporters, a tiny glycine, allowing this trap door to open. So the glycerol transporters, the aquaglyceroporins, have a larger pore. Now, I finished my medical training, was on the faculty, and never once considered that glycerol transport might have some biological significance. It just had never dawned on me. But the presence of the Aquaglyceroporin 3 in basal levels of skin is robust, to say the least. Very intense staining of the basal levels of skin. Now, one summer, several years ago, I was visited by some executives from the Christian Dior company. I don't know about you, but I don't get a lot of visitors from the Christian Dior company executive office. And as you can expect, they wanted to give me something but get something. They wanted me to give a lecture in Paris. And they were about to announce the release of a new product, something called Hydraction Skin Cream. It costs a fortune, 50 euros for a 50-gram jar. And their chemist had identified some small molecules that allegedly subtle increases the Aquaglyceroporin 3 expression in skin. Now, the notion that if you use enough of this, you'll look like this was not proven to my knowledge. And I'm pretty skeptical. And I should point out, I have no financial ties to Christian Dior. They offered some, but they had a lot of strings attached. My wife reminds me of that. It might not have been the best decision, but anyway, there you have it. Now, those of you who read French will see this is the back cover of the Marie Claire Beauty magazine. Visible hydration, spectacular results. And what's this, the Nobel Prize in Chemistry? I showed this to my mother, 80-year-old farm girl, now living alone in Minnesota. My mother saw this and she smiled and said, "Peter, I think you're finally doing something useful." Well, our mothers know us better than we know ourselves. I hope some of the other work is useful, but I think the Aquaglyceroporin 3 is going to have some important applications. It's present in red cells. We talked about the basal levels of skin. And in red cells, it provides the interesting function of glycerol transport, which I think every member of the transfusion medicine audience realizes is how we can glycerolyze cells for cryopreservation. So it's a functional importance in the transfusion medicine community. It also turns out to be the high-frequency GIL antigen, a very rare high-frequency antigen. This was work of Jean-Pierre Cartron and his group. And there are some rare individuals who are Aquaporin 3 null, and they seem to have no red cell manifestations of this defect. Their red cell glycerol transport is reduced, but they may have a defect in skin hydration. So there may be a negative phenotype here. Now, the Aquaglyceroporin 3 turns out to be really important in malaria. Because malaria, and I'll talk about this now in some depth, is an important clinical disease where the parasite will invade a red cell. A single parasite will grow and divide 2, 4, 8, 16, 32. 32 daughter cells will grow rapidly without the enzymatic machinery to synthesize glycerol. It's dependent upon the GIL antigen, the membrane glycerol transporter, which is also present in the vacuolar membrane and the parasite's own aquaglyceroporin for glycerol uptake. And so, as I said, here's an individual parasite. Here's 32. And this cell will break. 32 daughter cells are released. You can do the numbers. 32 times 32 is 1,000. 2 cycles 1,000. 4 cycles 1,000,000. 6 cycles 1,000,000,000. Horrendous fevers, high levels of toxic malarial cruds circulating through the circulations. So I'll just point to 2 points of this slide. We knocked the Aquaglyceroporin gene out of the Plasmodium berghei, this being the mouse variant of malaria. And what we found is that the parasitemia, the fraction of red cells that are infected, which normally goes up rapidly in about a week, took longer. The infection is diminished in virulence. The mice survived longer. Here are the mice injected with the wild-type parasite. They're all dead in 22 days. Those injected with the mutant parasites survive a bit longer. We haven't cured the disease, but it shows that it's important in terms of the virulence. Probably not enough to be a drug target. So there was a time in my career I wanted to work in world health, and I got very active in the laboratory, but I'd never forgotten that need to get involved in global health activities, and became the director of the Malaria Research Institute at Johns Hopkins, and had the great fortune to be part of our program as part of the NIH-sponsored International Centers of Excellence for Malaria Research. There are 10 centers worldwide, and we operate the one in Southern Africa. So we're working, as you can see, in Zambia and Zimbabwe, the former colony of Rhodesia, Zim and Zam as we call them. We have 3 different sites where field work is ongoing, getting parasites, looking for drug resistance, looking for insecticide resistance, getting a handle on the disease. So who gets malaria? And the answer is it's the small children of the developing world, and in sub-Saharan Africa most of all. Children like these little boys who in previous years would probably all have had malaria, but now can be protected, and if they do unfortunately develop it, can be treated effectively. But not everyone is so fortunate. Here's a little boy brought to the field station near death. He was in coma. His life was saved by Dr. Philip Tuma, shown on the right here, a Johns Hopkins pediatrician who's dedicated his life to working in sub-Saharan Africa. But the boy, while he recovered, he never regained his sight. He has a disconjugate vision there because he has sustained cerebral malaria, a horrific consequence of malaria. So we were made aware constantly of the 600,000 children who died last year of malaria, but there are several million who are left with kidney disease, lung disease, brain disease, and so the disease extent is much larger. So cerebral malaria has multiple components, but one part of it is similar to the brain edema that we defined in our studies with Aquaporin-1. Here is mouse specimens, normal mouse spec. Here's the basement membrane at the capillary interface with an astroglial end feet. Black labeling represents Aquaporin-1. And here's the specimen from a mouse treated with malaria developing cerebral malaria, these pockets of blister-like fluid all through the brain. And that combined with infarct leads to the damage that left this poor boy sightless. So just a little bit about our institute. Here we are, it's the surveillance team a few years back. It's amazing sometimes when we think how complex some diseases are, how simple intrusions can have such powerful effect. And malaria is such an example. By effective, aggressive treatment, Zambia converted in 2002, I believe, to the artemisinin in combination. And with aggressive follow-up, the rate of the infection declined. The Macha hospital adjacent to our field station had, in every previous year had at least 1,000 children aged 5 or younger admitted, with between 30 to 100 and some deaths. By providing effective treatment and following it up, we could knock it down. But there was a medical stock outage in 2006, and so the parasites returned right away and came back down. The introduction of insecticide-treated bed nets, which occurred in 2007, led to further decline in the amount of malaria present. So it's way down. It's about 97% below what it was, but it's not gone entirely. And so we know what will happen. If we cease our efforts there, it'll come back. So the problem is how to take care of malaria everywhere. And that's going to require a lot of geopolitical will, because borders do not stop malaria. You can make people cross borders with passports, but the mosquitoes that carry the malaria go back and forth. We're currently studying Anopheles gambia, the major malaria mosquito in Africa. And as you probably would not be surprised, they have their own cascade of aquaporins, 8 different aquaporins. Some are water selective, and these cause the mosquito to be able to jettison fluid rapidly after taking in a blood meal so flight can be accomplished. And there's some aquaglyceroporins, which turn out to be quite interesting in terms of the survival of this mosquito in harsh climates. Here we have, by RNAi, diminished the aquaglyceroporin 3 from the mosquito and treated the mosquitoes to exposure to 39 degrees, which is stressful for them. The survival time is shortened when the aquaglyceroporin 3 levels are reduced. Likewise, we looked at the numbers of oocysts per midgut. When the mosquito takes in a blood meal, the parasite crosses the midgut and forms an oocyst. And while the range of values is considerable, the wild-type mosquitoes had about 10 oocysts per midgut, whereas the parasites, the reduced expression of the aquaglyceroporin 3 had only 3. So it seems somehow this protein is important for the survival of the mosquito in stress and also confers the ability to form midgut cysts. Interesting, but how can we use this therapeutically? And that's a question I ask myself all the time. I'll end my discussion by talking just very briefly about 2 other aquaglyceroporins. These are now back to human and mammalian tissues. Aquaglyceroporin 7, which is present in fat, aquaglyceroporin 9, which is present in liver. So during fasting, we face a challenge. We run through our glycogen levels. How do we maintain blood glucose? And one of the early steps is the release of glycerol from fat. And it's clear from the work of several laboratories that aquaglyceroporin 7 allows glycerol release from fat, which is taken up by the liver through aquaglyceroporin 9, which is itself upregulated during fasting, allowing the maintenance of blood sugar, even to the late stages of starvation. This is work of Jen Carberry, who's now a faculty member down at Duke. Now, interesting, the aquaglyceroporins turn out to be freely permeated by certain heavy metals, notably arsenite. Arsenic trioxide is uncharged at neutral pH. And it is freely permeated, and of course, arsenic is something we're all concerned about. The groundwater and the public drinking water in the United States is very clean. But in some parts of the world, in particular eastern India and Bangladesh, where the surface water is contaminated with cholera, they put in tube wells to take groundwater up. It turns out in many locations, the groundwater is toxic with arsenic levels thousands of times above the acceptable level. The World Health Organization calculates 140 million individuals, such as this lady in Bangladesh, 140 million people drinking arsenic-toxic water every day with an epidemic of hepatocellular carcinoma. So the answer, of course, is the provision of pure drinking water. But what could this protein be there for? Probably the detoxification of arsenic, which all mammals must have faced during evolution. So in a simple study, again, collaboration with Søren Nielsen, here is hepatocyte decoration with aquaglyceroporin 9 antibody. Here's a knockout mouse. And here are feces measured after injection with arsenite. Wild-type mice injected with arsenite will excrete it rapidly. The null mice retain it. And of course, they succumb more readily. So the answer is it's probably a detoxifying agent for arsenic and other heavy metals in drinking water. Plants also have aquaporins. This slide I borrowed from Ralph Kaldenhoff from the University of Würzburg shows a wild-type Arabidopsis and an Arabidopsis engineered to reduce the rootlet expression of one of 35 different families of water channels. And it's enough to show that the plant compensates. It has a nice arborization by sending out more rootlets. Each rootlet with diminished water uptake, but more is enough to compensate. And of course, plants compete with each other for water during drought. So in closing, I'll just mention, I talked about the permeation of the water selectivity, the glycerol and nitrate and arsenite permeations of some subset members, the structural information, and some of the diseases where these are now been established to be involved. But the challenge is to do something useful with this. That's not so easy. Now, October's coming up. In 3.5 weeks, a few people around the world are going to be awakened early in the morning by a pleasant telephone call. And I'll share my experience in October 2003. At 5:30 in the morning, the phone rang. I answered, and it was a very pleasant Swedish voice. "This is an important telephone call from Stockholm for Professor Peter Agri. Are you Professor Agri?" I said, "I sure am." And it continued to explain that I'd be sharing the Nobel Prize in Chemistry with Roderick McKinnon. Rod McKinnon, like myself, is a medical doctor who's working on channels. He's up at Rockefeller. And they're going on and get to be introduced to the members of the committee. And I'm thinking to myself, do I tell them that I got a D in high school chemistry? Didn't seem like a good idea. And then I'm starting to imagine my high school chemistry teacher, Mr. Thornton, aspirating his cornflakes when he hears the news. I just held my peace and said thank you. And then he said, in 10 minutes there'll be a press conference in Stockholm. You better get ready for your day. So I sprinted to the shower. I was jubilant. And my wife, Mary, always calm, always organized, called my mother back in Minnesota, tell her the news. Mother listened thoughtfully. She said, "Mary, tell Peter that's very nice, but don't let this go to his head." She was still on that do something useful business, which is proper. It's totally proper. But you can't always choose the level of the celebration. Driving home that evening, I noticed that the Cut Rate liquor store was celebrating the announcement. And I'd like to defend my reputation. I'm not their best customer by anyone's measure. But here we are on the stage after the prize. And I guess any resemblance I had to Alfred Nobel changed when I took off the mustache. But now I've grown a beard. I don't know, I look like Santa Claus. There are 2 prizes I'd like to mention. The Nobel Medal, which goes to individuals, but it's for the entire work of a field. Really, these represent the advances from many laboratories. And it's great PR for science. It's the first thing on public radio they'll announce. Even Fox News will talk about it. It's amazing. The other prize here is my family. And I think that's something we all have. The lives of scientists are not easy. They're challenging, but they can be rewarding, but we rely upon them for love and support, and that makes all the difference. So careers also go very fast. I was just thinking it was 10 years since your report, and it's 20 years. Pretty soon we're in the later stages of our career, and we look back and we think about the science, and that was wonderful. But we also think back about the people we worked with, and that was really wonderful. The collection of friends we've had for our entire careers, they come with us in our minds and in our hearts. So the last slide I'd like to end with is something I've learned from the young people in the lab. Connie Liu, who just finished, she's now joined the Food and Drug Administration. My last scientist as I closed my laboratory. She taught me something I thought was pretty important. The character for crisis in Chinese, Wei Ji, is 2 characters. Wei, a time of danger, Ji, a time of opportunity. And when we think of the dangers of the world in terms of climate change, pestilence, disease, and we think in terms of science as the answer to these challenges, I think we all have lots of work to do and should be glad to have the opportunities to do that. And with that, let me thank the members of the Department of Transfusion Medicine here at the National Institutes of Health for the invitation to speak to you. And I'll be around. There's a reception afterwards. Thanks so much. Sure, I'll be glad.
Thank you very much for this great presentation, and we have a couple of minutes for any questions that you may have.
Thank you.
Yes.
Fantastic talk.
Thank you.
For more than one dimension. I have a lot of questions, but briefly, I would pose maybe 2. Are these Aquaporins, selective 1, 2, 3, developed more during the aging process, so that the aged people are not as sensitive to destruction of the water channels. And also, are they a specific site for each one of these units of channels in the immune privileged tissue, like the brain you mentioned, and also immune responsive tissue?
Well, first question, I believe there probably is a decline in many physiological pathways as we age. At 66, I can still go jogging, but I certainly can't run as fast as I used to run. My heart won't beat as fast. I can't move oxygen as fast. I won't get into the Viagra business, but you know, we basically compensate. And I think in some cases it may be a simple decline in the number of the transport molecules, but it's not been clearly established that the number of aquaporins counted in tissues reflects age. I think you're right, but it hasn't been proven. The second question about the specific locations, and I think that's where our colleague Søren Nielsen and our Norwegian colleagues, Ole Petter Offersen and their team in Oslo, did very beautiful work. And the answer is, it seems that they're very, very specifically located. The Aquaporin-1 protein, which is very important in the collecting duct, is clearly intracellular most of the time, and then it'll traffic to the cell apical surface. Mark Knepper and his team is working on that right now, doing very convincing work. It seems to rarely, if ever, go to the basolateral surface, whereas Aquaglyceroporin-3 and Aquaporin-4 may be at the basolateral surface. So they don't get mixed up. It seems that they're very specific. And I think that's held true with all the species that have been examined that I'm aware of. Did I answer your question?
Yes. Well, they could be also, they are more sensitive if they are in immune privileged tissue toward oxidation, as you somewhat suggested.
Well, that sounds like chemistry, which is not my field. My apologies to the Nobel Committee.
Thank you.
Thank you. Yes, please.
You mentioned several varieties of aquaporins. The question is the transport rate different in all these different types of aquaporins?
Yeah, the actual water transport rate is not easy to measure precisely. So the estimates must have some plus or minus 25% measuring error. But it appears that the aquaporins, the water selective ones, are all fairly similar. And there's some papers pointing that one might be more than another, but then they're often conflicting results. The glycerol transporting homologs have water permeability that appears to be less than the aquaporins, but they're also permeated by glycerol or urea and some other small uncharged solutes, including arsenite. So there are differences in the classes, but I'd not feel confident that I could tell you that Aquaporin-2 has a greater unit capacity than Aquaporin-1. They're both very large. And when you calculate the number of these channels, this has been done, I think one of Mark's early studies, the number of channels in the known water transport in the proximal convoluted tubules, it's enough to explain the massive water movement.
So glycerin is much bigger than
yes.
Water. So is there some flexibility in this ion in the channel aquaporin to accommodate a larger structure?
Yes. I probably went over it too quickly. There was a slide that I borrowed from Bing Yap at the Lawrence Berkeley Laboratory. So the aquaporin pore is about 3 angstroms in diameter, and the average diameter of water molecule is 2.8 angstroms. The glycerol transporter is missing a key histidine, which projects into the membrane and has a phenylalanine in its place. And the phenylalanine, this has been done by molecular dynamics simulation. Klaus Colton and his team in Illinois, Bert de Groot, Helmut Grootmüller, and Göttingen. The phenylalanine drops like a trap door, enlarging the pore. So we can explain it, and we can understand it. But you would think anything large enough for glycerol would let water go through indiscriminately, increased level permeation, and that is not found. So one question I'm often asked is there gating? Are these open and shut permanently, or are they flickering? And there's one member of the family I didn't talk about, Aquaporin-1, which turns out to have an ion conductivity, the only one. And the ion conductivity seems to be gated by pH, and it's rapidly flickering. So it may be that there's a gating that we can't measure in terms of water because it's electrically silent. That's about all I know about it.
You also showed the liquor stored. Does the alcohol goes to this channel, Aquaporin?
Yeah. Well, there was an effort in my laboratory to measure alcohol transport through the aquaporins, thinking that we could explain things like neural effects of alcohol. But alcohol is quite hydrophobic, and the barrier to diffusion is minimal. So it has very, very high level of permeation just through the simple membranes itself. So I guess we can't explain that.
Thank you for the inspiring talk and outstanding.
Thanks so much. Thank you.
I would have a question. The null variants of Colton, the non-functional alleles are rare, very rare. But variants of blood group proteins, including Colton, are relatively common. Do you see a scientific reason or interesting characterization of protein variants, Colton variants in the population? Like, where are the proteins going to? How could that be regulated?
Sure. It's something we've not addressed. I think identifying polymorphisms sometimes can be revealing. And oftentimes you get results that you can't initially explain, and it leads you in a new direction. There may be some functional difference in these polymorphisms that we don't know about. So I would hope someone would pursue that. It's too late in my career to do that. But I think here in this group there must be some interest in molecular genetics of even the Colton blood group antigens. And I do like the name Colvin better than Colton.
Yeah, interest is certainly there. I hope we'll also get the funding for the studies.
That's it. We've got to get the funding.
Yeah. If there is no additional question, then there is a reception next door in the National Library of Medicine, and we'll meet there. So thank you very much for this presentation. It was a great pleasure.